Is it a good time to start, Sandy? What do you think?
I think it's a good time to start now that Bob's here.
We got everyone we care, I guess. Okay, Andre, I guess that you get to introduce the guest.
Okay, great. It's my great honor to introduce today's seminar speaker, fantastic scientist, my former PhD mentor and still role model as of a scientist, James DeGregori. James got his undergrad degree from University of Texas, Austin. Then he moved to do his PhD studies into MIT with Earl Ruley, where he used retroviral gene traps, insertional mutagenesis to discover oncogenes and tumor suppressor genes. After that, he did his postdoc with Joan Evans, who has quite a few offsprings here at Moffitt Cancer Center. And when James established his own lab in University of Colorado, he initially started working on cell cycle deregulation and cancer within fairly kind of high quality, but fairly mainstream experimental effort. And at the time when I joined his lab in 2001, James started to think from fairly unorthodox angle of applying Darwinian principles of Darwinian evolution to resolve some of the puzzles of cancer initiation. And so it was like very fortunate for me to join him at this time, and that I continue working on these ideas and really now developed a multiple very cool lines of investigations. And initially it was all experimental work, but we kind of realized the importance of using mathematical models to get a deeper understanding of evolutionary biology. Unfortunately, we couldn't find mathematical collaborators when I was in the lab, but after I joined, James recruited a brilliant self-taught mathematician and very deep thinker, Andrei Rozhok, and they've made a number of very cool papers that are still mostly ignored in the cancer research community, but I think they're going to get appreciated more and more over time. So with that, I would like to pass the button and really looking forward to listening to about links between aging and cancer and sort of shattering the dug up erroneous idea that it's all about accumulation of mutations.
All right, thanks so much, Andre. Let me see, okay, wait, I meant to start at the beginning. That is close to the beginning. Let me get to there. Thanks so much. It's really wonderful to give this talk to investigators of the Moffitt. Not only do I have, Andre, a former student, but I've got even some of my old postdoc buddies are there. And a lot of great friends that I've made over the years. I almost feel like it's my second home. So the title of my talk is In the Light of Evolution, Why Do We Get More Cancers in Old Age? And I think most of you will recognize that the first part of that is from Dobzhansky's famous quote, that basically we really can't understand anything in biology except in the light of evolution. And I think the same is really true for cancer biology as well. By the way, this photo was taken when I was in Florida, so that's part of the reason I picked it. It's you might recognize it's a sunrise on the Atlantic side of Florida. I was visiting the Mayo in Florida. So I'll be talking today, I'll go into some theory, some of the in silico modeling that Andre referred to that I've done pretty much entirely with Andrei Rozhok. Then I'll talk about some mouse models, and I ended up deleting the human studies. Actually, as an evolutionary biologist, I've always felt that that if it's relevant in a mouse, it's relevant. I mean, to me, it hardly matters whether something is only in humans or not. In fact, I actually always have argued that the most important things will be relevant across animals, not just across even mammals. So I'll be really emphasizing the theory, and that was at the direction of David Basanta. So we'll start with this question. We know that 90% of cancers develop after the age of 50. And that's shown in this curve. This is SEER data. And you could see, oh, by the way, let me actually change my pointer. I'd like to have so everyone can see my pointer well, I hope. So basically, you know, what the sort of the theme today is that you don't want to, you know, age past 50. And because everything bad to us happens after 50. And this is kind of depressing, but true. And so you can see it here that your rate of cancer just goes up dramatically after 50. This is all cancers combined, leukemias, everything. And the real question is why? And to a large extent, the community thinks they understand this, the cancer research community. So they think that, well, this requires time. You know, that's part of the aging process, and cell divisions that happen over our lifespan. It's related to our exposures. And there's debate in the field about how much is due to cell divisions and how much is due to exposures, and as well as some influence of our inherited genetics. But pretty much, no matter how much people consider it's due to the cell divisions or how much is due to the exposures, the thought is this all comes down to one factor, and that's mutations. And so almost all the modeling that has been done up until this point has been basically focused on how do you accumulate a set of mutations. And it really has assumed that the factors like age and exposures are influencing this one parameter of sort of the evolutionary equation. But if we think about it, has evolution just been driven by the occurrence of the right mutations over time? And hopefully everyone, if you weren't all muted, would be saying a resoundingly no. Evolution's been driven by environmental change. And so if we think about these different evolutionary transitions that have happened over time, these have been really driven by drastic changes within the environment. So each of these periods is often punctuated by massive changes in the environment. You know, of course, we all think about the meteor that hit off the Yucatan 65 million years ago, but there have been other transitions. I think it was the Permian transition, where they think that there was a number of large volcanic eruptions that changed the gases, they changed climate, they changed a lot of factors. There were even periods where Earth was basically enveloped in ice. Huge changes in oxygen concentrations and so forth. And these changes drive the adaptation of species that can be adapted to the new environment. This isn't anything new. I mean, this is the way we understand evolution. And of course, even within these periods, there's environmental change driving additional change, maybe not at that dramatic level that you might get in between these periods. So that's how evolution works, but that's not how we see cancer, at least not how most people see cancer. And so still the current paradigm is that cancer incidence is limited by the occurrence of oncogenic mutations. And so basically modeling has just basically demonstrated that, well, if you add in enough mutations, you'll get a nice logarithmic curve, and you can make this curve look very much like that Sear curve that I showed. And that was the early work of Armitage and Dole and others, who basically concluded that you needed somewhere on the order of 5 to 7 events to account for cancer incidence curves. But we know that all cancers don't require 5 to 7 events. In fact, this number, which is still kind of unknown for many cancers, is quite variable. So if we consider these 3 different leukemias, CML, AML, acute myeloid leukemias, and CLL, chronic lymphocytic leukemias. CML, most diagnoses occur in the chronic phase, and we believe that this is driven by really one mutation, the BCR-ABL translocation. AML and CLL are thought to be driven by multiple different genetic events. And then we can consider different carcinomas. And these carcinomas, like colorectal, is thought to be driven by as many as 11 different driver events by the modeling that are the calculations that Martin Carena and colleagues have done. And going down to mesothelioma and to bladder cancers, where they're driven by far fewer numbers of mutations, with really only one discovered for mesothelioma. Now, we can take these numbers with a grain of salt, right? We don't really know the number of events because we don't really know how to integrate in the number of epigenetic events. We don't know how to really account for some of the larger genomic changes that are happening. But still, I think it's safe to say that the number of driver events that's required to form these different malignancies is highly variable. And that's my major point. And we also can consider that these different cancers are originating in very different stem cell niches. You can have these very drift-dominated epithelial stem cell niches, like for the colon or the intestines, where it's on the order of 10 to 20 stem cells that are physically separated, but you have, up to 10 million of these different niches. So huge numbers of total stem cells, but each one sort of isolated from the others to some extent. Or you can have intermixing pools like in the hematopoietic system, which are much more selection dominated. And it's thought that, for example, hematopoietic stem cells are numbered about on the order of 100,000 for humans. And basically they're pretty much intermixing. So it's to some level, even a stem cell in the bone in your big toe on one foot is in competition with the stem cell in the bone on your other foot, in the big toe of your other foot. So yet despite these huge differences in stem cell organization, large differences in the number of drivers required to form a malignancy, we see that really, if you normalize all these curves for these different cancers and you simply set their maximum at 1 and then use their minimum, which tends to be 0 at some point at least, you will basically see their shapes are incredibly similar. I was incredibly struck when Andre showed me this curve, because basically it shows that all these different cancers have an incredibly similar shape of incidence curves. And this curve even looks like the curve for clonal hematopoiesis. As most of you know, clonal hematopoiesis, by the time we're older, like in our 70s, many of us will have clonal hematopoiesis where we have large clones driven by sometimes oncogenic mutations and sometimes by unknown events, but they're clonal and they could even come to dominate our blood system. And this curve of incidence, which we're pretty confident is largely driven by single events, also replicates the same curve. So that's the mystery. So what Andre set out to do, and by the way, this is a different Andre Roshak, who, by the way, Andre Marusik introduced me to, so he can take some credit for hooking us up in the first place. And what Andre did is he developed a Monte Carlo model that tests the multi-stage model of carcinogenesis, which I'll refer to as the MMC. And the model is relatively simple. He starts off with a pool of cells. And for much of what we're doing, he's using 10,000 because that's thought to be the number in a mouse, but it's on the order of 100,000 in humans, and he varies this pool number. And then cells with that pool will either stay quiescent or they divide. And the size of this pool is dependent on a known curve of the size of the hematopoietic stem cell pool within humans, or really within most species, which obviously when we're born is going to be relatively small, and it increases largely proportionally with body size as we reach maturity, and then stays pretty constant. And their division rate, he initially set this to be based on additional data that he derived from telomere shortening numbers and other data, which estimates that you would start off with a much higher rate of division at birth, and obviously during ontogeny, and that division would slowly plateau in such that by the time you're reaching maturity, it's going to be a relatively lower rate of division that you maintain for the rest of your life. He also did a constant rate too, and really it didn't make a lot of difference. But the cells then divide, and then they accumulate mutations, and they can either accumulate a driver mutation or not. And that's dependent on the mutation rate that he set, which he also varied. And here's the important point. When they accumulate a driver, their cell somatic fitness is updated. And it's updated based on two different methods, two different sort of modes. The first one is the standard multi-stage model of carcinogenesis, which is that basically oncogenically initiated self-renewing cells always have a greater stable somatic fitness advantage over normal cells. So basically the oncogenic event confers some advantage, and we will vary that amount of that advantage. That's the standard model. But then he had another model that's basically what we call DS, MMC DS, which is basically differential selection. And he has two different modes of this. One where you get a proportional change where in youth, basically for the first half of your life, up to about 50 years, the effect of the mutation, this oncogenic driver, will be negative. And then it'll transition over to becoming positive late in life. So where it's basically the selection against the mutation in youth and selection for the mutation late in life, with the curve that he basically derived from the aging curve for humans. And then he basically varied the magnitude of these effects proportionally for youth versus old age. Or he had another mode where basically, if it started out more negative, it ended up being less positive afterwards and so forth. And finally, he had a third set of experiments where he kept the mode of this the same, except he basically changed when this decline occurred, essentially to model animals with different lifespans. So you can imagine these might be mice and dogs or something down here, and these might be humans, and these might be elephants or something like that. And so basically, once the somatic cell fitness is updated, then the cells are now in competition with each other because the pool size will be constant, and the cell will either leave the pool or stay in the pool. And that's going to be basically somewhat stochastic, but the probability of staying in the pool will be dependent on the fitness of the cell.
Can I ask a brief question here before we move on?
I think someone's not muted.
Yeah, I asked if we are allowed to ask questions in between. Or
Oh, I'm sorry, I didn't hear that. Yes, you absolutely can. Is it Heiko?
Yes, hi, how are you?
Hi. Yeah, go ahead.
So if you go back one slide, please. So the shape of the DS function, the differential selection, you said is adapted from the shape of the age distribution in the SEER data.
Yes.
So my question, if you do that, then probably you get results that look like what you see in the age distribution in the SEER data.
Yeah,
So
no, exactly. The curve was based on survival data for humans, not cancer specific. So the curve is based on just human survival over 100 years.
Do you think this curve could also be derived differently from some kind of biological feedback?
We certainly could have. I mean, essentially, that's how he made the curves below. Is he just assumed different, assumed a similar shaped curve for humans, like mice under ideal conditions, for example. Like for example, if you have mice in the lab, almost all of them will be alive at 18 months. You'll start to lose some by 24, and then by 28, 29 months, you've lost most of them. So that same sort of decline, but we didn't change that model with other parameters, but I get your point.
Okay, thank you.
Oh, sure. Yeah, please, anyone who wants to ask a question, feel free. I've also got a slide smack in the middle where I basically stop to answer questions because I've learned that the attention span of a human is about 20 minutes. So this is the results of some of the modeling. So first in the MMC, the standard model, he simply changes the fitness value of the driver mutation. But there's no differential mode. And what you could see for one mutation clones, he basically almost uniformly gets early life malignancies. And then the more mutations you add, you can start shifting some of these curves to the right. And what I'll note is that you can find conditions where you can get late life exponential curves independent of the number of mutations that you pick. So you can make the MMC work. You can find parameters that make it work. But I think what the lesson I'd like to leave from this is that you then have to really get very imaginative. You have to imagine that the 4 mutation clones, each of those mutations is conferring a very, very small fitness advantage. While like for a 1 mutation clone, you would actually have to confer an incredibly small fitness advantage to get it work. And so you almost have to have magical thinking to imagine this sort of perfection in terms of that each oncogenic variant is driving a very different sort of fitness effect on the cells. In contrast, if you use the differential selection model, and it really works for either model, you could see that you could pretty uniformly delay cancers till late in life. And that's of course not surprising because the events are disadvantageous early in life. And you can also see that there's still variability. In other words, the number of drivers does still matter in terms of the event. And that does present still a problem, which I'll deal with in just a minute. So what I'm not showing you, by the way, this was all published in eLife. Most of what I'll tell you in the rest of the talk is unpublished. This was published in eLife last year. So the differential selection model is also more robust to changes in the target stem cell pool size and the mutation rate. And so a lot of this led Andre to think about this in terms of, how is it that you almost magically get all these different cancers with all their different parameters so uniformly clustered late in life? And so what he figured is that, well, you can imagine that if you have some animal and there's some somatic mutation that could cause early malignancy that would impact fitness. I mean, we're talking about leading to the death by cancer, but you can imagine this, maybe it would just slow the animal down and more likely mean it's going to be eaten by a predator. There's a lot of ways of thinking about this. It doesn't necessarily have to be lethal by itself. It might affect its ability to reproduce, for example, but where it's affecting its fitness. And you might imagine that 2 different drivers are required for this other malignancy. And because it's going to require 2, it's going to take longer to happen. It's going to basically be a lower odds event that's going to require a first clonal expansion driven by the first driver, leading to the second driver clonal expansion. If that was the case, then you would expect very different germline selection for cellular mechanisms suppressing somatic evolution. That will depend on the effect of the particular mutation on overall reproductive success. So you can imagine this first mutation would essentially be more dangerous to the animal, and there'll be greater selective pressure to suppress it. More tumor suppressive mechanisms will need to be evolved. But these other events would require less of that. Basically, it would require selection against them, but it wouldn't need to basically limit it forever. And you can also imagine this will vary even between animals. Because, for example, a mouse, even a wild field mouse, which isn't going to likely be alive after a year, won't need to suppress 5 mutation cancers, for example, or 10 mutation cancers that are with 10 driver mutations. But a human or an elephant or a blue whale would. And so you can imagine this is going to affect differences between species. And so by combining this with our sort of standard way of thinking about this differential selection, which is inherent to the adaptive oncogenesis model, which I'll get to in a minute, I think we can explain how we get this sort of convergence of cancer rates and even leukemia rates to a very similar time late in life. And so essentially what I'm arguing is that this is really, these common curves are the product of natural selection. And that, of course, shouldn't surprise anyone. And why this is the case, I think, is pretty obvious to most people. And then you might be thinking, yeah, but what about childhood cancers like leukemias, testicular cancers, and lymphomas that happen in younger people? And I don't have time to go into them in great extent, but I think this exception to these late life patterns will often be, 1, associated with modern lifestyles in developed nations. That's definitely the case. For example, there's some breast cancers that occur in sort of, even 30-year-old and 40-year-old women. And we know that there's pretty good evidence that that's probably due to changes in parity and breastfeeding and other modern type predisposing factors, and even things like obesity. So natural selection hasn't had time to catch up. The same may be true for childhood leukemias. At least Mel Greaves has argued that they're way more common in industrialized nations. Another factor is they tend to be rare. So almost all these, as you can see from that SEER curve that I showed, the vast majority of malignancies, particularly life-threatening malignancies, are relegated to the latter half of a century. And finally, they can be associated with pathogens, because pathogens have their own evolutionary agenda. So our ability to evolve tumor suppressive mechanisms to prevent them is going to be limited by the basically evolutionary battle with those pathogens. So of course, the 10,000-foot reason is sort of summarized here. Why is it that most cancers are relegated late in life? I'd say it's the same reason that most disease, and I'm graphing here heart disease, but you could pick your favorite bad thing that happens to us late in life, and the curves are actually quite similar. There's really minimal selection against diseases of aging beyond the age where most individuals would already be dead by other causes. So this is a curve of European survival about 15,000 years ago. I think if we elect our current president for a second term, I think we'll get a very similar curve in the United States. Sorry, just got a tickle in my throat. Sorry. And we can see that, hold on one second. But we can see that there's very little overlap with the survival of this population, and based on fossil data in cancer and heart disease that's occurring in modern populations. And so the idea would be that cancer, that humans and pretty much most species in the wild have evolved to limit cancer incidence to the point that it's having a relatively minor effect on their fitness. That doesn't mean that during evolution it didn't have a major effect. In other words, I think the very reason that cancer is off to the right is because it otherwise would have had a major effect on fitness. And we can see this in modern animal populations, particularly ones that have been impacted by human activity, where now there are some populations where cancer is having a major effect on fitness. And that's because they're no longer well adapted to their environment, or they've been put through some evolutionary bottleneck or something like that, where they become susceptible. But evolution has acted to push these diseases to the right. And you might think, well, there is some overlap. I mean, there's some reduction of fitness. Why wasn't this pushed further? And probably the answer is because there's a cost to tumor suppression. There's a cost to maintaining our soma for so long, such that further maintenance would come with a cost. And that cost would be reduced ability to produce offspring in youth. So there's basically limited resources, and those resources need to be invested in a wise manner. And so if we think about this, explains, and this is the curve I was referring to, Heiko, is this is a standard human survival curve for death by any reasons. And this is why life is not linear. Actually, your chance of dying in your 20th year is no different than your chance of dying in your 30th year. And it's barely higher in your 40th year. It is significantly higher. But really, you can see that your chance of dying in your next year starts accelerating rapidly. And basically, after the age of 60, not only do we go downhill, we pick up speed. And your chance of reaching your next birthday becomes less and less as we get older. And this is, of course, because this is where the investment is made. This is where the somatic investment is made. And it's not like there's just a point where that investment is turned off, but that investment wanes as we get older because our chance of contributing to the next generation has gone down. So this raises the question, well, then how does lifespan evolve in the first place? Now, I probably don't need to explain this to this crowd, but I just love this example. And I love it in part because my son did the artwork, but I also love it because I think it's such a clear example. And I know there's caveats associated with it, but let's gloss over those for now. So Steve Ostad did this really nice study where he was looking at the Virginia opossum, and he's actually looking in Georgia, but he looked at survival in this, or senescence, the markers of senescence in the mainland population versus on this nearby island that's been physically separated from the mainland for about 5,000 years. So basically, these have been isolated populations. They are still the same species, but the ones on the island aged about 50% slower. So if you looked at markers of senescence like tail fibroblast and stuff like that, that a 2-year-old possum from the island had younger looking cells than a 2-year-old from the mainland. And the reason for this is pretty clear. So you might want to think about, what is it about being on the mainland versus the island? And you might guess that, well, on islands, predators, larger predators particularly, have trouble surviving with smaller populations. They require much more area to basically find their prey and persist and so predators often go extinct on islands. And in fact, the island is pretty much absent for non-flying predators. So there's no coyotes, there were no wolves, and there were, and some of the other animals that would normally prey on the possum. It's not like they have no predators, but they have reduced predation. And this is sort of graphed out here. You can imagine on the mainland that with age, their chance of surviving largely because of predation goes down dramatically. And so there's only selection for maintenance up to the point that's going to maximize reproductive success. And so that's going to wane as they get into ages where they would be unlikely to survive. But when those possums ended up on this island, this curve could be shifted. And so now there's less predation and other extrinsic hazards. And so you can increase this germline selection, and such that you can basically extend sort of the fitness curve. And what this is going to do is it's going to lead to changes in cancer incidence, because basically, as we extend the maintenance of the tissue fitness, we can push cancer incidence further and further off to the right. So I think if we really want to reduce cancer incidence, we need to figure this out. So, and I think this can help explain things like PTOs paradox, which I think people are familiar with. So you can have species, as large as the blue whale, which has 10 to the 7th fold more cells than a mouse, lives about 50 times as long, but has no greater problem with cancer. In other words, I don't think cancer reduces the fitness of a whale any more than it reduces the fitness of a mouse. And this is true just across mammalian species by great work from, Amy Bodie and colleagues. So what this then raises the question is, well, how does that work? Well, from a sort of traditional mutation accumulation problem, this creates a real problem, because how do you have 10 to the 7th fold more cells and have to maintain those cells for 50 times longer and still avoid cancers? And what I'd argue is that I don't think that maintenance of the tissue fitness is the entire story, but I think it's a good part of the story, because essentially the whale would fall into an extreme example of this, because it can really, once it's an adult, it can really avoid predation. It can really avoid extrinsic hazards. Of course, modern times accepted thanks to humans, but for most of its evolutionary history. And thus it's able to basically invest in its soma much longer. And as I'll show you in a bit, investment in the soma is itself tumor suppressive. So then what does this investment in tissue maintenance during youth and the waning of this investment in old age have to do with cancer? And this is the theory that my lab's been developing since the days of Andrei Marusik in my lab, adaptive oncogenesis. So this theory really has two key components. I should say model. I don't think one can call your own ideas a theory. I think someone else has to call it a theory. But the first part of this is that high stem cell fitness opposes somatic evolution and favors the status quo. So just like if you have a population of animals, plants, bacteria, or whatever, and if that population is well adapted to its environment, then it will tend to basically be under the influence of stabilizing selection. In other words, any different parameter, any parameter you might pick, phenotypic parameter for that population, if it's changed, it's going to tend to reduce the fitness of the animal, not increase it. So you can think about, birds. If you have a population of adult swallows, they're going to have wing lengths that are, within some range. There's going to be variability, but not a ton of variability, because a bird that had much shorter wings or much longer wings would be less fit than those that have, within the right range of wing length. So more is not necessarily better, and certainly less is not necessarily better. But what this means is that even though a malignant genotype might have a higher fitness, if getting there means going through intermediates that have lower fitness because of these changes in parameters, that transition will be difficult and it'll be rare. And I'm using words like that on purpose because it doesn't mean it's impossible. A 30-year-old who does everything right, who doesn't smoke, eats perfect diet, and runs, 5 miles every day can still get cancer. But just he or she is much less likely to get that cancer than a 70-year-old who smoked their whole life. So it's a matter of probabilities. And natural selection, I believe, has shaped our tissues for a very similar effect. So we have evolved stem cells that are well adapted to their tissue niche. And by the very fact that they're well adapted, they're going to tend to resist change. So change will tend to be disadvantageous, or at least it will not be advantageous. And thus it'll make this transition more difficult because basically there's less room for improvement. But we don't stay young and healthy forever. Either we get old or we do things like we smoke or we have other exposures that reduce the sort of microenvironment of our tissue because the fitness of those stem cells is dependent on their niche. And so in fact, I think it's largely microenvironmental changes that reduce the fitness of this population of cells. And that's additional modeling by Andrei Rosak showed that from an earlier paper of ours, that you really need changes in the niche. You need changes in the environment that reduces the fitness of the whole population of cells. And if that's the case, Now, even a cell that has a wild type genotype will find itself less adapted to its environment. So you can imagine a lung stem cell that evolved to be in a nice, healthy young person's lung, and it's now either finds itself in an old lung or in a smoker's lung, will no longer be adapted to that environment. And so you'll select for mutations that provide an adaptation. Now, these mutations aren't necessarily, these adaptive mutations aren't necessarily going to lead to a malignancy. In fact, actually, we're learning nowadays, a lot of them don't. A lot of adaptations are non-malignant. They can even help restore tissue function, but it's going to increase the odds that you do get malignant evolution. I should mention that a lot of these ideas, actually, a friend of mine, Niles Eldridge, his books and writings majorly influence some of these ideas, if that's not already obvious. In fact, we recently published together. If you want to check it out, it's available online. So to sum this up, natural selection has acted over millions of years to maintain animal bodies to the extent that maximizes reproductive success. Of course, that's no surprise to anyone. I mean, that's not our idea. That's been since the days of Medawar and Williams and others. That's been the thinking. But what we're saying is that maintaining tissues in their youthful state is potently tumor suppressive. So just the act of having a healthy tissue will impede somatic evolution. Somatic evolution is less limited during that period by the occurrence of the mutation and more limited by the fact that the mutations are going to not tend to be adaptive. But when we get aging and tissue damage, now you select for change, which can sometimes lead to cancer. And now this evolution will be more limited by the occurrence of the mutation, because now the more mutations you have, the more likely one of them is going to be adaptive and oncogenic. So you might be thinking, so what is this evidence for differential selection for somatic mutations dependent on context? I showed you theory, I showed you computational modeling, but what's the actual evidence? Well, it turns out there's actually a lot out there, and a bunch of it's been from my lab, but a lot of it shown in green is from other labs. Really great examples over the years. Some of the earliest examples that I can think about were in the skin by, oh my God, I'm suddenly blanking on his name. But yeah, Doug, it'll come to me. But anyway, he showed that you could select for p53 mutations following UV damage to the skin, but all the way through different models, including an aging type context. And I'm going to just show you a few of those. And so one of those is from Curtis Henry when he was a postdoc in the lab. And what Curtis wanted to ask is, could we ask, and this was following up on work actually that Andrei Marusik started in the lab, but Curtis now wanted to know whether we could actually interrupt this evolution. So what he did is he took cells from young mice, and these are all young stem cells. He introduced this RAS oncogene into a small fraction of these cells, basically such that some minor fraction would have RAS and be GFP positive. So that's the first thing we're doing different than other people, is that we're only introducing it to a small fraction of the cells. And then he put these into mice. And first off, these mice were busulfan treated. He worked out a protocol that unlike radiation, so you have to keep this in mind. A lot of people claim that their oncogenic events are inherently advantageous because they think, whoa, I put it into a young animal and it expanded like crazy. Well, they're putting it into an irradiated animal, which is not a normal animal. It's not presenting a normal environment. But what he showed was with busulfan treatment, it's only mildly inflammatory. It doesn't tend to reduce total cellularity in the bone marrow. And even the inflammatory cytokines within 2 to 3 days go back to baseline. So it seems to be only a brief insult that's quickly sort of resolved, and it specifically deletes hematopoietic stem cells. And when we do transplant with these, most of the cells remain of the host age or type. And so when he puts these cells into young mice, or old mice that are transgenic for this IL-37, which is anti-inflammatory, he can then ask what happens to those cells. So keep in mind, the cells that he's putting in are all identical, same pool of cells. The only thing changing is the tissue environment. And when he puts young cells into a young mouse, and he basically sees that these oncogenic variants don't provide any fitness advantage. The cells stay rare at the same frequency that we put them in. It's really hard to say that they're eliminated because we're putting them in so little frequency. Some of them end up undetectable, but we can't really say that they're negatively selected. But we can say they aren't positively selected. In contrast, when he puts these cells into old environments, and this is one of the wonderful things about working with aged environments, we always get high variability. And that's probably because, you know, aging is not a uniform event. Even 5 litter mates in the same cage that are 2 years old, well, if you just looked at different aging parameters, you'd see variability in those aging parameters. But we see variable but pretty consistent expansion of these cells. What was really striking is just by blocking inflammation, he could largely prevent that. And then he asked, well, why is this? And what he showed is that young cells have a lot of sort of what we would call their normal fitness parameters. And so this is, think of this like the wing length of the swallows. It's what we would say evolution has favored. This is the evolved level of, for example, STAT5 signaling, but he looked at a number of gene expression parameters and other things to basically determine, well, that's the amount that you would normally see in these B cell progenitors. But in the old case, a lot of these were reduced, and we see reductions, for example, in STAT5 signaling. Same thing for different transcripts like MYC levels and different nucleotide synthesis genes. But what he found is that if he had RAS in these old cells, it would largely restore these parameters to somewhat normal levels. And so we could see why RAS would be adaptive. If we assume that this is reducing fitness, and if RAS is restoring the levels of these parameters, then these cells would be more fit than these cells. And that was indeed the case because these expanded relative to the non-NRAS transformed competition. But importantly, when he blocked inflammation, the fitness of the cells was not 100% restored, but it was apparently restored enough that we no longer selected for this NRAS mutation. So that room for improvement was taken away. And we're starting to see a lot of examples of this in the literature now for humans. I'm not showing you all of it, but I already mentioned clonal hematopoiesis before. Beautiful work from the Martin Karainen colleagues from the Sanger group in England, showing that you get these different mutant clones colonizing, for example, the human esophagus with age. And actually really cool recent work from the colon, which I think is really nice because, as I mentioned before, not all somatic evolution is bad. In fact, these appear to be downregulated NF-kappa B signaling, and it seems to be adaptive within the colons of people with ulcerative colitis. And if anything seems to be restoring more normal colon morphology. And so essentially what we're seeing is that there's a ton of somatic evolution going on in humans, and most of the somatic evolution is relegated to older ages. So there's that same exponential increase in older ages. So to kind of summarize this up is that you can imagine we accumulate genetic damage throughout our whole life. Epigenetically, it seems to be a lot of it front-loaded when we're dividing a lot. But depending on what stem cell population you look at, it's either sort of a linear increase with life, or it's more of this sort of front-loaded increase followed by pretty linear afterwards. But either way, you have a lot of genetic damage that happens before you get old. And yet, despite this, at least that damage that changes phenotype is going to be acted on by stabilizing selection, such that you maintain the phenotype of normal. The evolved phenotype is favored. But late in life, when natural selection has no longer acted to maintain the soma of the animal to the same extent that it did when it was young, when that sort of maintenance wanes, now with that environmental change, you can get positive selection for new phenotypes that are adaptive to the new environment. And some of those phenotypic changes can be adaptive. And we also know that by modulating the environment, we can suppress that. So this is where I was going to stop and answer a few questions. I have a second half, but I was going to answer a few questions if people have any, just to give people a mental break.
James, this is a great presentation. Very, very thought-provoking as well. It's great. So I'm still trying to figure out how to phrase this, but you know the work of Inigo, Martin Karainen and company about the fact that we have a prevalence of mutations in healthy tissues that is something that people are just beginning to look at.
Right.
How do you put that in the context of your work? I think it's very compatible, but I'm just trying to figure out whether this adds something to it.
Yeah, no, I mean, I'm a huge fan of Martin Karainen and all, and not just because he's Spanish. So, but basically, I mean, what you see in the esophagus is really interesting because the majority of clones, the dominant clone are in Notch1 mutations. And I actually wrote a little paper with a student in my lab about this, that if you look, these Notch1 mutant clones expand. So right now, my esophagus, because of my age, is probably 50% Notch1 mutant. And yet my odds of getting esophageal cancer, because I'm a never smoker and I don't drink excessively, is really, really low. And yet over half of my esophagus is populated by what we would have previously thought was an oncogenic clone. So what that says is that there's a great favoring of somatic evolution as we get older, but it's not necessarily dangerous to us. Mm-hmm. And I've argued that those clones are what I would call sort of decoy fitness peaks. So you can imagine if that Notch mutant clone is restoring fitness, by restoring fitness, even if only partially, it's gonna lessen the odds that you would then have selection for a malignant clone. Because these may not be completely normal, but they're normal enough. In fact, actually, one of the things that this group, Phil Jones and colleagues, noted was that morphologically, these look normal. The esophagus, despite being riddled with clones, looks normal, seems to function fine. So I think a lot of somatic evolution that we have during our life, much of it's benign, but it can be malignant. And so actually, I think these different papers fit with that very nicely. The one exception maybe is the work from Louisa Moore in the endometrium, where they are seeing clones that happen earlier in life, like in the 30s and 40s. Now, but again, those may not be malignant. And so given that still endometrial cancer is relegated to late in life, I think, in fact, that's one of the things I noticed with the Notch mutant clones, those start coming up in your 40s. Mm. But it's the dangerous ones like the P53 mutant clones, those are relegated to mostly the 70-year-olds, keeping in mind that their numbers were small. But in other words, I think natural selection doesn't have to prevent somatic evolution. It just has to prevent somatic evolution that's dangerous. It's going to reduce our fitness. That makes sense?
Yes, it does. We have time for one more question. And I think that, Joel was asking for the chance to ask something.
Sure, sure.
Howdy, so good to see you. I wish.
Hey, Joel, how you doing?
Around Paris as we did before.
Yeah.
Completely with you, as always, that selection ceases to keep us alive once we kind of outlive our Darwinian program.
Sadly.
It seems like a key feature of your novel theoretical construct is that the whole organism is evolving along some sort of adaptive landscape, the wing of the bird.
Yeah.
That you're suggesting this is achieved by each somatic cell within the organism also evolving along some sort of adaptive landscape within the beast. So this idea then that somehow selection maximizing the fitness of an individual cell is also creating the team optimum. Am I interpreting that correct?
Yeah, to some extent. I mean, it's still obviously evolution is happening. Essentially, we are but vehicles for our germline, just like for every animal. And so natural selection has basically maximized the chance of that vehicle is going to deliver success in terms of offspring that are themselves going to survive to adulthood. So, for example, evolution has favored lungs, where the structure of the lung and the structure of the whole organization with the stem cells is, you know, and I hate to use the word maximize because nothing is maximal, but optimizes that fit between stem cell and niche, so that those stem cells are relatively competitive. And I think there's really good examples of this in the skin from Seiichi Ogawa's group. Oh, wait, no, it wasn't Seiichi Ogawa for the skin example. It's a Nature paper, and I actually wrote a news and views on it if you want to find it. But beautiful work showing that the skin cells, the basal cells, are in competition with each other. And if one of those cells gets damaged, it basically loses some of its attachments. And thus it's more likely its neighbors are going to force it out. Now, it doesn't kill the cell. That cell just now is more likely to differentiate up the layer of the skin and be a useful skin cell, but it won't be contributing to the long-term maintenance of that tissue. So I think the best work of this has been in Drosophila mechanistically, but now we're starting to see it even in mammals, where cell competition is essential for maintaining youthfulness. And so when they disable cell competition, they actually get premature aging. And if they promote cell competition, they get longer lives. But of course, there's a cost to that. You know, you can imagine having better cell competition, eliminating every cell at the drop of the hat would become expensive. So it's a balance. Does that answer your question, Joel?
Sure, you bet. No, appreciate that, James. Thanks.
Sure. Next time we'll do it over a beer.
Absolutely.
All right. Any other ones before I move on? All right. And now I'm going to get to more data. And this, everything from now on is unpublished. So this is a study that was done by Kelly in my lab. Kelly just graduated. And it was a collaboration with Eric Pietrus's lab. And Eric is was actually Kelly's co-mentor on this. So the question really was, so I showed you that inflammation was necessary to promote selection for oncogenic events in that one context of the B progenitor pool, but is it sufficient? And so what Kelly was considering is that there's this critical dynamic in hematopoietic stem and progenitor cells where they self-renew at a certain rate and they differentiate at a certain rate. And she noted that many inflammatory contexts will, like aging, radiation exposure, chemo, will inhibit the self-renewal and promote the differentiation. In fact, this is actually a critical factor. So when you get an infection, which is of course inflammatory, this promotion of granulocytes is essential for your survival. It's called emergency granulopoiesis. So this burst of differentiation every time you get infected or you get damage to your tissues is actually a critical evolved mechanism. But it can become chronic under some circumstances and leads to basically reductions in the fitness of the stem cell pool. And she also noted that a lot of adaptive oncogenic events do the exact opposite. So a lot of AML-associated events actually promote self-renewal at the expense of differentiation. And then she thought, well, if I'm going to model this, what should I look at? And she used IL-1 beta as the model for inducing inflammation because Eric had done a lot of previous work on that, and it's critical for this emergency granulopoiesis. And she used CBP alpha loss as the critical adaptive event to test because she knew that these are inhibited by a lot of these oncogenic events. They act to inhibit CBP alpha, which is known to promote myeloid differentiation. And what she showed is that if she makes chimeric mice, again, using that method that Curtis had developed, which is basically this really mild method of introducing cells into a mouse. And she introduces a small fraction of cells that have either deleted or wild type CBP alpha that are YFP marked, together with competitor cells into congenic recipients. And then she basically either treats them with IL-1 or not. And she asks, what happens to those cells? So the first surprise was that in contrast to published work, losing CBP alpha by itself was not advantageous. And like I mentioned before, that's because previous work was done where they irradiated the recipient. In fact, they even caused deletion using poly-IC, which is itself inflammatory. So essentially they had seen an advantage, but they were using a very inflammatory context. But in our model, which is non-inflammatory, we did not see this advantage in any of the compartments we looked at. These are all early progenitor compartments, by the way. You don't need to worry about what they are exactly. But in the presence of IL-1, we see potent selection for the cells that have lost CBP alpha. So they're adaptive specifically in the inflammatory event. Again, context-dependent adaptation. And what she then went on to show is that she could even see this in vitro. And in vitro, we can really look at these parameters and take it out of the sort of complex environment of a mouse. But first off, you can see that IL-1 reduces the ability of these stem cells to maintain themselves. At the same time, if you compare this to this, increasing their differentiation. And in both cases, this is partially reversed by losing CBP alpha, where you better maintain the stem cells and you reduce the differentiation, such that over time, you select for the cells that have CBP alpha loss in vitro. Now then the question is, how is it doing this? How is it conferring a fitness advantage? And what she did then is she did the same competitive transplants, same treatment, and then she sorted out this early hematopoietic stem and progenitor population, MPP3s, and looked at them by RNA-seq. And not only did she look at the cells of the different genotypes that we were most interested in these different contexts, but we did RNA-seq on the competitors. And I'll show you a bit of that data as well. But first, you'll notice that IL-1 induces a lot of changes. So it makes a lot of genes go up and a lot of genes go down relative to the vehicle controls. Not surprising. what was really striking is when we lose CEBP alpha, we majorly reverse these programs. So a lot of the genes go up, fail to go up in the absence of CEBP alpha. This in itself is really interesting because it's essentially saying that CEBP alpha is the master transcription factor necessary for emergency hematopoiesis, this emergency granulopoiesis. This, you know, when you get an infection, you produce a ton of these granulocytes. You need CEBP alpha to even do that. And also, many of the genes that went down failed to go down if you don't have CEBP alpha. It's not 100%, but many of them do. And when we looked at the gene expression profiles, we saw some really dramatic effects that jumped out of us. So IL-1 beta decreases stemness genes because it's promoting differentiation. And you could see that quite clearly on one of these enrichment plots. And that's largely reversed by CEBP alpha loss. It restores those stemness genes. In contrast, IL-1 beta increases myeloid gene expression just dramatically. And again, that's largely reversed by CEBP alpha loss. And then we want to know, well, do we see this in sort of clonogenic assays? And indeed, when she plated out these progenitor cells, if they were CEBP alpha deleted, they would survive longer. So just deleting CEBP alpha, at least in vitro, was enough to confer self-renewal. But in the presence of IL-1 treatment, we saw the synergistic effect. We massively increased clonogenic potential with that combination. Because essentially what she's seeing is we're now taking away the negative effects of IL-1. So those are being negated, and you're essentially revealing the positive effects of CEBP alpha loss. Now, this was the cool experiment, and I was really glad this worked because basically to do all those competitor RNA-seqs doubled the cost of it. And I hate when I have an experiment. I shouldn't say do an experiment because I'm not doing them. But, you know, I hate when I pay for an experiment and get nothing out of it. But we did in this case. So now instead of looking at the actual cells that with the different genotypes, we're looking at the identical genotype competitor cells. So these are the cells that were sorted out that were all got treated with IL-1, but they were either in competition with CEBP alpha deleted cells or against wild type cells. And what she saw, saw this big enrichment. If the cells were in competition with deleted cells, they were even more enhanced for myeloid differentiation and even more depleted for the stem cell genes. In other words, they did even worse than they would have just by IL-1 treatment alone. So you can imagine that the CEBP alpha delta cells in the presence of IL-1 aren't just winning because they're failing to do these, they're actually making their competitors even worse. And we don't know how they're doing it. It could simply be that there's a certain amount of demand for making granulocytes. And if these guys aren't doing it because they can't, because they lack CEBP alpha, it puts greater demand on these and they're forced down that lineage even more. So to sum this up, IL-1 impairs stemness and promotes myeloid differentiation, all of which is reversed by CEBP alpha loss. And the CEBP alpha knockout confers a fitness advantage to HSC only in the context of IL-1, not in the standard context. So what essentially is happening is IL-1 is causing HSC dysfunction, and this may serve as a driving force for HSC clonal evolution by selecting for variants that are basically restoring the fitness of the cells. And that's pretty much summarized down here. So I should ask my host, David, I'm just going to fly over the lung cancer stuff so we can get to the end because we're at the end of the hour and I think we're going to start losing participants. So I'm not going to go into this data, but I just want to show you that, you know, we know that lung cancer is age dependent. We know that the lung changes physiologic. Yeah.
Yes, just 1 second. So of course, you know, like if you can skim anything you want, but and some people might have to leave in a couple of minutes, but we allocate an hour and a half for the whole thing because we know people like to ask questions. So
okay, I can go through this in just a few minutes.
Sure. Yes.
Yeah, and it will be recorded on YouTube, so people will be able to
go back to it.
Yeah.
Okay, sounds good. So I guess the first thing what I'll point out is, so this is not surprising, right? Here's smokers, never smokers, and lung cancer. Same thing for women. Obviously, smoking is a bad idea if you want to avoid lung cancer because there's a massive increase in your risk of lung cancer with smoking. So whether you get lung cancer is determined by your smoking. But when you get it, and this is seen in this log-log plot, that's determined by your age. So even smokers largely avoid lung cancer until they're 40 or above, just like never smokers largely avoid lung cancer until they're 40 or above. So what's changing in old age? Well, we know there's a lot of changes in the physiology of the lung, the reduced basically capacity to breathe. There's also a lot of immune changes, which have basically come out to increased levels of inflammation in the lungs of an old person. And so we want to ask, well, are these changes in old age sufficient to select for oncogenic events? And so we're using one of Andrei Marusic's favorite oncogenic events, the EML4 ALK. And we're not introducing this fusion gene. What we're actually introducing is CRISPR constructs that create breaks in the ALK gene and in the EML4 gene, which are both on the same chromosome, just like they are in humans. So I should mention this fusion event is most common in lung cancers of never smokers, and it's an age-associated event. So the average age of individuals that get EML4 ALK positive or ALK positive lung cancers in general is about 67. And it's when you make these CRISPR cuts, you get a spontaneous flipping of this piece of the chromosome to generate the EML4 ALK translocation. I'm actually amazed this works in this model, but it does. It's actually quite efficient. And we're doing this either in young or old mice. And first I'll mention that we can measure whether these events occur, and this is from the results of the Ventura lab, simply by looking at phospho-3 positive foci, which are relatively rare in the absence of putting in this EML4 ALK inducing virus. But their frequency of these events in different tissue sections is pretty similar for young and old. So we're able to induce the translocations at similar rates. Now we can ask, are they adaptive? And when we look at that, we can see there's a big difference. So each dash is a mouse, and most of these mice have none of these adenomas when we section across the lung, but some of them might have one or maybe two adenomas. But the old mice all have adenomas, and they tend to have multiple adenomas each. So there's a big increase in the frequency of adenomas when we induce these events in older mice. Now, key thing is, can we do anything about it? And so we then used another transgenic mouse, which I don't have time to go into how it does it, but alpha-1 antitrypsin is anti-inflammatory in these backgrounds. It does it by blocking the neutrophilic protease reactions. And you could see that they—
James, can I just ask briefly, are these mice immune competent?
They are, and I'll get into that. And that's, you might notice the rag knockout over here on the right. I'll explain something on that. Yes, they are immune competent. These are wild type BLAK6 mice. Okay. That's the beauty of this method. We do not have to have anything special about the mice. We're just inducing the CRISPR by putting adenoviruses that have the Cas9 and the guide RNAs into the lungs of the mice. Okay. So just young and old mice, perfectly immune competent. We get them from the NIA, National Institutes of Aging. And we could see that, again, we're getting a big increase in the numbers and the size of these adenomas in old relative to young. Most young don't have any. And if we block inflammation, we can largely prevent it. Basically, we get a young-like pattern. Now, in reference to what Sandy was just referring to, we want to know, well, could this be due to the decline in the adaptive immunity in old age? And what we find is it's not, because if we take young mice, but we take away their adaptive immunity by making RAG1 mutant, we get a basically very similar pattern of these adenomas. So I wouldn't say there's no role for the immune system, but I don't think the immune system is playing much of a role at this early stage. I think the immune system really kicks in when the tumor starts getting disruptive, but I don't have good evidence for that. But there's actually others that have speculated on that similarly. So we think that blocking this inflammation is changing the landscape to basically restore, at least partially restore, a more youthful phenotype. And so to look at that landscape, we did proteomics with our collaborators, Travis and Kirk. And we find that, first off, you might not be surprised to find that there's a lot of changes that happen in old. These are different proteins in old relative to young. Some go down, some go up. But having the alpha-1 trypsin, antitrypsin inhibitor expressed all the time doesn't change a lot of this. So some aging ain't a changing. But importantly, there is a subset, I'd say it's roughly a third of the proteins, seem to be reversed back to somewhat normal patterns. But now the question is, so we know that we reversed a lot of the oncogenesis. So are these the key ones then that are involved in reversing that oncogenesis? And we look at what these are. Some of them, not surprisingly, are very pro-inflammatory proteins like S100, A8, and A9. They go massively up in the old background. So they note this is a split scale. And they're basically brought down to close to background by the AAT transgene shown here in blue. So white is young, gray is old, blue is anti-inflammatory. We also see a number of proteins involved in tissue remodeling, like MMP9 and elastase, that go way up in old and brought back down to baseline by the anti-inflammatory transgenic. So we think that there's a partial reversal of the phenotype that seems to prevent this oncogenic adaptation. I should mention that instead of using a transgene, we're now using antibodies to block either specific immune cells or specific cytokines. And I'm pretty confident that we're seeing effects that we can reverse this adaptation, but I want to see it in another independent set of mice before I start presenting it. So essentially, we're arguing that oncogenic lesions preferentially expand in the age backgrounds, and that reductions in the inflammatory environment associated with old age can markedly decrease this degree of oncogenic clonal expansions. And I'll note that aspirin and anti-IL-1 beta treatments can reduce cancer risk in humans, so that's consistent with this. And also AAT deficient humans actually exhibit a several-fold increase in lung cancer risk. So while we may not be able to prevent most mutations, or at least many of the mutations that can happen as we get older, we can modulate their impact by manipulating the microenvironment. And I'd argue that we don't necessarily want to block all clonal evolution. And I think this very much fits with what you guys are doing at the Moffitt. We want to direct that clonal direction in a more benign fashion, sort of like direct cells up those decoy fitness peaks instead of having them go up to more malignant peaks. And I'll end on one note, and that's that because you can't not mention COVID in these times. And so this is curves, again, for cancers shown in the colored bars, but also for heart disease and other things. And you can see, again, getting older past 50 is a really bad idea because everything bad starts to happen to you. I guess we all want to get old. We just don't want to age. And you notice this is COVID-19 death rates. Basically, it's the same curve. After 50, your chance of dying from a COVID infection goes up dramatically. So I think the take-home message should be that efforts to prevent and treat cancer should really converge with similar efforts to prevent and treat other aging-associated diseases. That we need to maintain our soma because a better soma, shoring up our body's soma, is the best way to improve our defenses against malignant actors, whether this is a malignant cell like we in the cancer biology community like to think of, or whether it's a virus like coronavirus. And we need to leverage also markers of declining soma, like clonal hematopoiesis, as an ecosystem check for our soma. So who should get the intervention, for example? And so with that, I'll first thank people. So my lab has been fantastic, and I've just added a couple new people to the lab. Former people, I talked about their work, like Curtis and Hannah, Nate, and Andre. All the artwork was done by my son, Michael, and he did the artwork in my book. And so a lot of those pictures were stolen from my book. Great support now largely from the National Institutes of Aging. And I mentioned the collaborators along the way. I should mention we got the CBP alpha knockout mice from Dan Tenen, who's a collaborator of ours. He's been great. And a lot of local collaborators. A lot of the bioinformatics was done with Andrew Goodspeed. And this is just a picture of a recent picture of the lab that doesn't have the newest people, but pretty recent. And with that, I'm happy to take any questions.
Thank you. So I know that some people were actually deferring the questions until now. So Philip, do you want to start? Philip Alstruck?
Yo, hey, hi, this is Phil. James, thanks
Hi,
for
Phil.
this talk. Awesome. Yeah, well, you can say. Sorry, I don't have my headphones, so hopefully you don't hear an echo. I mean, so there's this, we are, right, I feel for the first part of your talk, okay, it's the soma, but then there's always, and I don't really have a great idea about this, but I have some intuition. The aging of the immune system in and of itself, and it's starting its mixture. I mean, I think you hinted it in terms of like more toward the adaptive, or toward the innate versus the immune,
Right.
and its checkpoints, and how that is right, always the first line of defense, as Sandy usually puts it, the first treatment that the cancer has already grown out of, and how does this play into this kind of lifetime of aging and the payoffs, in a sense. That's something I would be a bit more fluffy, but interested in. Hear your thoughts.
Yeah, no, this is the beauty of calling something simply like, the context environment, because when I mean the environment, I mean the environment total. So that's not just like the immediate, surrounding cells, but that's the immune environment. And so you can imagine that for a cell to be adaptive in old age, it's going to need to be adaptive to a lot of the environmental changes, I think, are immune changes, whether it's by being more inflammatory, but that could also include being reduced adaptive immunity. My bias based on our work, because we've done with multiple of these different models, we've done like RAG mutant mice as comparisons. And we've never seen that losing T and B cells is sufficient to allow for clonal expansions. I'm biased towards, and I've seen other people like Michael Caron write similar things, to the idea that you can imagine, like if you look at Martin Carena's work in the esophagus, if our esophagus becomes half oncogenic variant driven, and I imagine that some of those variants will be, immune stimulatory, then they could be immune stimulatory. But I think as long as they're not disrupting tissue function, you're not getting the second signal that you need for immune reactivation. You're not getting those inflammatory signals. So I think it's really, I would say, when the tumor starts busting through the basement membrane, when it starts basically causing trouble, then you're going to get inflammation. And at that point, I think you start getting immune selection. And that should also go down in old age, because we know the immune system is perturbed in old age. That's not what we've been studying, but I absolutely think it plays a role because really when we're looking at cancer incidence, we're not looking at benign malignancies. We're looking at the final aggressive cancer that's causing a problem.
Exactly, but in CHIP, we have an opportunity to somehow correlate these in an earlier stage. So going back to that.
And I'm willing to bet, and one of you guys may be able to do this, I bet if you took all clonal expansions that are seen late in life, and you asked whether, like all CHIP, you can find everything from these esophagus and uterine and all these different papers, and you asked, is there selection against well-presented epitopes, new epitopes? I will bet anyone a beer that there's not. But when you get to a full-blown cancer, you will see it. In other words, I bet at these early stages, I'm willing to bet that the immune system's pretty agnostic to these early clonal expansions.
Oh, great, thanks.
Yeah.
Yeah, maybe, I know you have a question. Looking for the mute button, maybe? Okay, I have more questions, so if maybe doesn't come back in a second. Sandy, do you have any yourself?
I do, yeah. Great talk, James, as always. Very insightful. Some cool new stuff at the end there in regards to the lung work. And that kind of intrigues me a great deal, because obviously in the standard sort of progression of many cancers, lung in particular, there's normally a polyp or a kind of, what do they call it?
Early adenomas, yeah.
Like a nodule stage,
right?
And so it's I don't know if we'd even call it an adenoma yet,
right?
Because it's not cancerous, it's just right? And so do these models that you're using allow for that sort of emergence of that stage, or does it sort of circumvent it completely?
I'd say that's actually what we are studying. We're looking 6 weeks after introduction of the event. I don't think there are additional events involved at this point, because we will see multiple of, in some ways they look almost like bacterial colonies, because you get these sort of, they're not all exactly nice balls, but they're clearly one localized from a single event type expansion. So I'd say that's what we're mostly studying. We actually have had trouble. We tried to have some of the mice go out to see if we had changes in carcinoma. Unfortunately, we're starting with old mice. And when you do that, most of the mice tend to die of other things before they die of their carcinoma, because normally this carcinomal development takes 4 to 5 months. And you can imagine if you're starting off with 20-month-old mice and you've already manipulated them, you just tend to get mice keeling over. So we haven't answered the progression problem, but we definitely have answered this sort of early expansion problem.
Yeah, that's cool. So, and it has some relation to some work that I've done with Trevor Graham's group, where we're looking at kind of colorectal cancer in those early stages, so adenomas and then progressing. And we see that there's a class of adenomas that are sort of separate from those adenomas that appear coexisting with carcinomas. So there's a progressive group of adenomas and a non-progressive group.
Yeah.
And I just wonder if you'd seen some separation there.
You know, we do. In fact, I should have brought this up, but I pulled it out, but it will be in our paper, which we hope to submit by the end of the year. But the adenomas, I think I know what you're talking about. There's AAHs that are in lungs. So they're adenomatous associated hyperplasias. And they're much smaller, and they're just sort of like, and they are clonal expansions. But when we look at those, we see a mild age dependence of those. But really, it's that continued expansion towards an adenoma that we found was so age dependent. So we only saw about a 2-fold difference in the formation of AAHs between young and old, as opposed to that big difference in terms of adenoma progression. So what I'd probably argue is the vast majority of AAHs are not going anywhere. And in a young person, they're really not going anywhere. Or at least I could say young mouse. But yes, so that we did look at an earlier stage, and there was much less of a difference at that earlier stage.
Cool, thanks.
Yeah.
So I made Parag Katira a panelist, so he can ask the question himself. Parag, are you still there?
Yes, thanks, David. Give me one second. Yeah, there we go. Again, this was a really exciting talk.
Thank you.
My first question, which I posted actually, was related to, so clearly aging is there, right? And you have this trend with aging and cancer incidence. And the model seems to really nicely capture that trend. Yeah. The question is, it's not universal though. I mean, you have a difference between male and female, you have a difference between ethnicities. Right. And how does this fit into that context? And then the immune system part also kind of adds to that same question, because depending on where you are, where you grow up, your immune system is differently triggered or more active, less active, because you're more infections. And so how does this correlate across different ethnicities, environmental conditions, and so on? Right?
I'm very interested in that question. I think it's a great question. So first off, male versus females. Most cancers are leftward shifted in men, but so is our aging curve. In other words, sadly, we're going to die, on average about 3 to 5 years sooner. So if you ask, what fraction of, old age homes are loaded with women, centenarians are heavily biased towards women. And there's a simple reason for that. And every time I do this, I give a talk like there's some guy in the audience goes, wait a minute, I can still reproduce, when I'm 80. And I'll go, not if you're dead. And so for most of our evolutionary history, and this is the case across mammals, even non-mammalian species, males tend to have higher mortality in youth. And they have higher mortality because, for example, competition for mates, fighting, and so basically males have been selected for early robustness. Even muscle mass takes a huge amount of energetic investment, because basically a male is like, if I don't breed early, evolutionarily, I'm toast. And of course, not thinking anything, right? This is what natural selection has done. So males are more front loaded in terms of our fitness. So we decline sooner. So I think that the leftward shifted curve of cancers is just concomitant with that leftward shifted decline in our fitness. Now, I think across ethnicities, and I think that's hugely important. And I do think it probably has a lot to do with what sort of pathogenic environments we grew up in. Because you can imagine that surviving an infection in youth is way more important than some change in your cancer risk when you're 60. And so I think most of the selection has been on an immune system that's optimally tuned to prevent death from our reduced fitness from infections early in life. But that's going to differentially affect our cancer risk later in life.
So is that going to increase or decrease the cancer risk?
I'm sorry?
That's going to decrease the cancer risk, though?
Well, it could decrease or increase. So let's say that you have a more inflammatory response because over evolutionary time, your ancestors were having to deal with, we're having to deal. So in other words, you're tuned to be more hyper responsive to, let's say, bacterial infections because your ancestors dealt with more of them. And I'm totally hand waving at this point, but now when you're older and you're more likely to become chronically inflamed as an older person because of it. So for example, we know that centenarians are enriched for genotypes that tend to be less inflammatory. But there's no indication that that would have been advantageous because there's no evolutionary advantage to living to 100, right? In other words, that was so rarely attained that there wouldn't have been selection on that. So you might imagine that might be a rare genotype because it wasn't the best genotype for surviving through youth. But I do think that our ancestral differences in terms of the pathogens we encountered or other threats could influence how we manifest, later states such that could either be enhancing or decreasing cancer risk.
Just beautiful. Lots of stuff to think about it.
Yeah, for sure.
I think we might have time for one more question, and Adam McLean is interested in asking. Adam?
Hi, Mr. Adam McLean.
Oh, okay.
Thanks, that's lovely work. I'm curious, so you showed for the emergency granulopoiesis, right, these very dramatic gene expression changes downstream. So I'm curious in turning that around and thinking about what is the role of the non-evolutionary component on this context-dependent oncogenesis? Like, how does the transcriptional state of a cell in these altered environments, affect risk? And do you think about that? Do you worry about quantifying that component?
Yeah, no, I think, first of all, I'd say that the emergency granulopoiesis in itself probably doesn't increase risk substantially. So work from Eric Pietras and Emmanuel Passague, when he was a postdoc in our lab, showed very nicely that if you take away the IL-1, you will reset and the hematopoietic stem cells return to the normal state, their normal fitness. And so it appears to be, of course, that's what we evolved to do. We evolved to respond to a threat and then go back to a normal set. I think where this becomes a problem, is when you have chronic inflammatory conditions like old age. And it's also known that, for example, people that have arthritis have an increased leukemia risk. So even chronic disease during youth can have this effect. So, so I think that the selection has been very strong for a very good emergency granulopoiesis program because it's essential for you to survive an immediate threat. But there's been this effect that it could increase selection for adaptive events when it's in its chronic state. Am I answering your question? I don't know if I lost track of your actual question.
Hey, James?
Yeah.
Could I ask one question? Of course, this is not relevant to me because I'm so young, but just
of course.
So how does this fit into the classic experiments that were done in the 50s with initiation promotion models of carcinogenesis? Have you kind of integrated that or thought about that?
Yeah, I think a lot of you and I agree, a lot of the promotion is probably acting, in fact, that was even shown with the TPA models, is that it was inducing differentiation. So the promotion may be selecting for, events that were induced by the initiator, selecting for those events such they would expand out. And, some of the, even experimental models have kind of shown this, is that just inducing, like p53 mutations in the skin is not enough. You really need the UV exposure. And so in some ways that's the promoter. So I do think in some ways that it's compatible with those ideas. Of course, we're talking about things like aging and smoking and other things like that, essentially being the promoter.
Thank you.
Sure.
Cool. I think there's more questions, but we've been going on for a while. So I guess that, we can always reach you out by email or maybe Zoom later, but you must be tired. And I think some of us need to go to their Zoom meetings now.
Yeah.
So
and feel free to tweet me. You could tweet at me. You can email me. However you want to contact me is fine. I'm faster on email though.
You're one of those.
Yes, that's because I'm over 50.
So am I. Well, thank you very much. Very thought-provoking.
Oh, thanks so much. It was a delight. Thanks for the great questions as well.
Okay,
Yep,
see you
thanks,
guys.
James.
Thanks, Sandy.